Hello friends, welcome to some biology class. Friends, the term complement refers to a set of serum proteins that cooperate with both the adaptive and the innate immune system to eliminate blood and tissue pathogen. The complement system is a complex group of more than 30 glycoproteins that interact with one another in catalytic cascades. Today, we shall discuss about the classical pathway of complement activation. The classical pathway is considered the part of the adaptive immune response since it begins with the formation of antigen-antibody complex. These complexes may be soluble or may be formed when an antibody binds with the antigenic determinants or epitopes present on the cell membrane of various microbes. Only complexes formed by IgM antibodies or certain subclasses of IgG antibodies are capable of activating the classical pathway. The formation of an antigen-antibody complex induces conformational changes in the non-antigen binding Fc portion of an antibody molecule. This conformational change exposes a binding site for the C1 complement component. The C1 component is a macromolecular complex consisting of one molecule of C1q, two molecules of C1r, and two molecules of C1s. This figure shows the composition of an C1 complex. This is a macromolecular complex consisting of one molecule of C1q, two molecules of C1r, and two molecules of C1s. C1r and C1s are serine proteases, and the C1q component is itself composed of 18 polypeptide chains. These 18 polypeptide chains are organized to form 6 collagen-like triple helical arms which have globular heads. These are the globular heads which must bind with the CH2 domain of an antigen-bound antibody molecule. This is the surface of a pathogen. These are the antigenic determinants, and these are the two IgG antibodies that have bind with the antigenic determinants to form antigen antibody complex. As already mentioned, the classical pathway of complement activation is considered part of the adaptive immune response because it begins with the formation of antigen antibody complex. This figure shows various intermediates of the classical pathway up to the formation of C5 convertase. This is a C1 macromolecular complex. These are the components C1r and C1s, and these are the C1q globular heads. This is the surface of the pathogen. These red dots are antigenic determinants or epitopes, and these Y-shaped structures are the antibody molecules. And this part is the non-antigen binding Fc portion of the antibody molecule. So when the globular heads bind with the non-antigen binding Fc portion of the antibody molecule, then only a stable interaction occurs. So each C1 macromolecular complex must bind by its C1q globular heads to at least 2 Fc sites for a stable C1 antibody interaction to occur. Friends, serum IgM exists as a pentamer of basic 4-chain immunoglobulin structure. Circulating non-antigen bound IgM molecules adapt A planar configuration in which the C1q binding sites are not exposed, but when the pentameric IgM binds with a multivalent antigen, it undergoes a conformational change to form a new configuration in which at least 3 C1q binding sites are exposed. Because of this, an pentameric IgM engaged in antibody-antigen complex can bind with the C1 complex, whereas the circulating non-antigen bound IgM cannot. The membrane-bound C3 convertase hydrolyzes C3 component generating two fragments. Smaller fragment C3A is an anaphylotoxin and the larger fragment C3B is enzymatically active. A single molecule of C3 convertase can generate over 200 molecules of C3B, thereby resulting in tremendous amplification at this stage of the classical pathway of complement activation. C3b molecules play three important and different roles. First, C3b molecules bind covalently to the microbial surface, providing a molecular tag, thereby allowing the phagocytic cells with C3b receptors to engulf these tagged microbes. This process is called as opsonization. Second, C3b components also bind with the Fc portions of the antibodies participating in soluble antigen-antibody complex. These C3b tagged immune complexes are bound on red blood cells or phagocytes by C3b receptors and are either phagocytosed or conveyed to the liver where they are destroyed. Finally, some C3b molecules bind with membrane localized C4b2A enzyme to form the membrane bound trimolecular C5 convertase complex. The function of C5 convertase complex is to cleave C5 component generating two fragments. Smaller fragment is C5a and the larger fragment is C5b. As you will see, C5b form membrane attack complex along with other complement components such as C6, 7, 8 and 9. Therefore, C4b2a3b is the C5 convertase of the classical complement pathway.